This is Module 3, Transcription. In this video, Prokaryotic Transcription, I'll describe some of the features of transcription that are unique to prokaryotes, specifically bacteria. To remind you from the last video, transcription is the synthesis of an RNA molecule using DNA as a template. So the substrate for RNA polymerase is DNA. The addition of the first ribonucleoside triphosphate, ribonucleoside, guys, not deoxyribonucleoside, does not require the use of a primer. And then subsequent ribonucleotides are added to the 3' end of the growing RNA molecule. These are the three major similarities between prokaryotic and eukaryotic transcription. Now we start to differ a little bit. The transcriptional machinery that binds to RNA in a prokaryote or a bacteria consists of an RNA polymerase, one type of bacterial RNA polymerase, and another licensing protein called a sigma factor. RNA polymerase in bacteria consists of 5 different subunits, 2 copies of an alpha subunit, 1 of a beta, 1 of a beta prime, and 1 of an omega. And so to create this protein molecule, 4 different genes are involved. So the alpha, the beta prime, and the omega. These DNA genes are transcribed into 4 different types of RNA, alpha, beta prime, and omega. And then alpha has a high rate of transcription, meaning that gene is made into RNA much faster, double the rate of the others, because we need 2 copies of that subunit, not just 1. When these RNA molecules are translated into proteins, we get the RNA polymerase. It is a protein in what's called its quaternary structure, meaning it consists of 5 different polypeptide chains that are each individually folded, and then those 5 folded proteins associate and come together to form this one major enzyme. That is the core RNA polymerase enzyme. It actually can't work unless it has its additional protein called a sigma factor. Once the sigma factor joins to the core of the core RNA polymerase, it forms what's called the holoenzyme, which is an active enzyme capable of binding to a promoter and beginning transcription. All types of transcription, bacterial or eukaryotic, consist of 3 different stages, but what's happening during each stage is a little bit different between bacteria and eukaryotes. Those stages are initiation, elongation, and termination, just like in DNA replication. Let's start with the initiation stage. Bacterial promoters are really simple. Bacteria are nice, simple organisms, and so they keep it simple for us. All bacterial promoters are essentially, not exactly, but fairly similar, and all have the same consensus sequence. There's 2 different, very specific DNA sequences that must be present on a DNA strand in order for it to begin, or in order for it to serve as a site for the beginning of transcription. At the -10 position, so this would be 10 base pairs upstream of the start site, so towards the 5' end of the non-template strand. That's the direction we're going. At this position, there is a sequence called the Pribnow box, which was named after the individual who discovered it. This is always TA-TA-AT, almost always. And then 35 bases upstream of the start site is yet another consensus sequence responsible, or that is also considered to be the promoter, which is TTG-ACA. You don't have to memorize the specific order of these nucleotides, but you should know what a Pribnow box is. What do I mean when I say consensus sequence? So this is an example of 4 different bacterial promoters. And this is being shown the -10 region. So that's the TA-TA-AT. So here it is, TA-TA, and then it should say AT, right? Oh no, wait, this is the other one. Oh, this is the complement, okay. Anyway, this is just one example of a consensus sequence. This is not actually the promoter, but this is just what consensus sequence means. So here would be 4 different sequences in 4 different organisms, 4 different species of bacteria. And then what you see is a lot of commonality between these 4 sequences. And so basically the average appearance of a nucleotide gives you the consensus sequence. And so in each of these, we have 3 T's and only 1 A, so the consensus would be T. Here we have 3 A's and 1 C, so the consensus is A. Here we have more ambiguity, and so what this Y means is that it just needs to be a pyrimidine of some kind. What this R means is that it just needs to be a purine of some kind. And the CG notation basically means cytosine and guanine are equally likely to occur at this position. So this is a consensus sequence, the average agreed upon sequence for a particular site. Promoter sequences in bacteria are consensus sequences. They are very, very similar. Across multiple species, they're often identical. And then some species have very small differences in their promoter regions. For example, I've worked with E. coli and I've worked with cholera in the past. And what I tried to do one time was genetically engineer cholera bacteria to contain an E. coli gene. The problem is I wanted that gene to get expressed and made into a protein. And what I found out through much trial and error was that the promoters between E. coli and Vibrio cholera differ by 1 base at the -10 consensus sequence. So this is A in E. coli, but T in cholera. And just that 1 base pair difference made it so that the protein was not expressed very well in cholera. The E. coli protein was not expressed very well in cholera. So there's a consensus sequence is the -35 upstream of the start site and a -10 upstream of the start site. The RNA transcript does not contain any of this promoter sequence. This is only found in the DNA. Okay, elongation in bacteria is, again, fairly straightforward and fairly simple compared to eukaryotic elongation. This is done by RNA polymerase, but it has to bind its sigma factor first. And so here we have the core 5 subunits, the RNA polymerase subunits, that associates with the sigma factor to form the holoenzyme. And then finally, this whole unit can bind to the promoter. And it's binding to the -35 and the -10 sequences in the promoter. Once that happens, the holoenzyme has a little bit of helicase activity and binds to that region and unwinds the double-stranded DNA, breaking hydrogen bonds that keep them together. Again, RNA polymerase does not need a primer to start building an RNA molecule from its template. And so it just right ahead tossed a G in there, which is opposite this C. And then each time a nucleotide is to be added, 2 phosphate groups are broken off of that nucleotide, which allows for, which generates the energy required to actually create this bond over here and add this nucleotide to the growing chain. Bacterial transcription consists, so bacterial termination also proceeds in similar ways to eukaryotes with a couple of differences. In bacteria, there are 2 different ways RNA polymerase can be told and signaled to stop transcribing RNA. One is a row-dependent termination, which uses a protein called a row factor, and the other is the row-independent termination, which uses secondary structures in RNA to signal RNA polymerase to stop transcribing. So first, let's look at the row-dependent method. Row is a protein that binds to a site near the 5' end of a growing RNA chain. Row is like a little Pac-Man guy. He actually starts to move up towards the 3' end. And when RNA polymerase bumps into the terminator sequence in the DNA, so there is a signal in the DNA that tells the RNA polymerase stop here, RNA polymerase will pause. And this gives a chance for Row the Pac-Man to catch up to the replication bubble. When it does, it uses helicase activity to unwind the DNA-RNA hybrid and then stop transcription because this protein is pulling RNA off of the DNA template. Termination by bacterial row-independent terminators is somewhat similar in that a hairpin is formed, but there's no protein involved. There's no protein involved at all. And so a row-independent terminator contains an inverted repeat. So this is what an inverted repeat is. So this is essentially a mirror image complement to itself. So here you see CCG, this is GGC, and so it's a mirror image of itself. And then immediately following these inverted repeats, there has to be about 6 adenine nucleotides for this to qualify as a termination signal. These inverted repeats are complementary to each other. And so when they're transcribed into RNA, they will associate with each other and form a double helix at just this region. This causes RNA polymerase to pause in folding to stabilize. And then once that finishes, the RNA transcript is pulled off of the DNA template. The formation of the hairpin actually destabilizes the complementary base pairing between the DNA and the RNA in the replication bubble.